Hello everyone, I'm Priyanka and today we will talk about sterilization. Sterilization is making a substance free from all microorganisms, both in vegetative and sporing state. Spore is a reproductive structure that is adapted for dispersal and surviving for extended period of time in unfavorable conditions. Spores form part of the life cycle of many bacteria, plants, algae, fungi, and some protozoa. Let us discuss some terms used in sterilization. First, we have disinfection, the destruction or removal of all pathogenic organisms capable of giving rise to infection. Disinfection does not affect spore state organisms. Next, we have antisepsis. The term is used to indicate the prevention of infection, usually by inhibiting the growth of bacteria in bones or tissues. This is done by the antisepsis, chemicals or disinfectants which can be safely applied on the skin or mucous membrane to prevent infection by inhibiting the growth of bacteria. Next we have germicidal pesticidal agent, those which are able to kill bacteria. Next we have bacteriostatic agent, only prevent multiplication of bacteria but they remain alive. Talking about cleaning, important preparatory step before sterilization or disinfection by removing soil and other dirt. Decontamination, the process of remaining an article or area free of contaminants including microbial, chemical, radioactive and other hazards. Now let us discuss the agent or method of sterilization. So we have two methods here, physical method and chemical method. Talking about the physical method or agent, we have sunlight, drying and heating. In heating we have further two type of heating, dry heat and moist heat. Dry heat sterilization include flaming, incineration and hot air oven. Talking about the moist heat, pasteurization, boiling, steam under pressure. Next we have filtration, use of different candles, asbestos pads, membranes. Then we have radiation and ultrasonic and sonic vibration. Next we have chemical agent like alcohols, ethyl isopropanol, tetrachlorobutanol, aldehyde, formaldehyde, dyes, halogens, phenols, surface active agents, metallic salt, uses of gas like ethylene oxide, formaldehyde. So, so firstly we will discuss the physical method of sterilization. First we have sunlight. Action primarily due to UV rays, however, effects vary due to places. For example, in tropical countries, the germicidal effect is better than four-season countries. Bacteria in water are readily destroyed by sunlight. Next, we have drying. Moisture is essential for growth of bacteria. Drying in air has delicious effect on many bacteria. However, spores are unaffected. Therefore, it is not really unreliable. Next we have heating, most reliable method of sterilization and should be the method of choice. Further we have two type of heat sterilization, that is dry heat and moist heat. First of all, let us discuss the factor influencing sterilization by heating. First we have nature of heat, that is dry or moist. Second important factor is temperature and time. At what temperature we are heating and what is the duration. Next we have number of organism present, characteristic of organism, that is species, stain, sporing capacity. Next we have type of material from which organism have to be eliminated. The killing effect is due to protein denaturation, oxidative damage and toxic effect of elevated level of electrolytes. Killing effect of moist heat is due to denaturation and coagulation of protein. Next we have thermal death time, TDT. Thermal death time is defined as the minimum time required to kill a suspension of organism at a predetermined temperature in a specified environment. Thermal death time is inversely proportional to temperature. TDT is increase in presence of organic substance, protein, nucleic acid, starch, gelatin, sugar, fats and oil. So next we have dry heating. In dry heating, first we have flaming, that is heating over fire till they become red hot. Instruments like points of forceps, spatula, inoculating loop and wires. Inoculating loop is better dipped in disinfectant first before flaming to prevent patching. Next we have incineration. It is a process that involves the combustion of organic substance contained in waste material. Items, contaminated clothes, pathogen, pathological materials, P.V.C., polyethylene, etc. However, polystyrene will emit black smoke, hence should be autoclave in appropriate container. Next we have hot air oven. Hot air oven is electrical device used in sterilization. The oven use dry heat to sterilize article. Generally, they can be operated from 50 to 300 degrees Celsius, that is around 122 to 572 degrees Fahrenheit. There is a thermostat controlling the temperature. This is the most widely used method of sterilization by dry heat. Items, glassware, forceps, scissors, scalpel, all glass syringes, swab, liquid paraffin, dusting powder, fat, grease, etc. Material should be properly arranged to allow free circulation of air. So this is a hot air oven. Talking about the precautions, glassware should be dry, oven should not be overloaded, articles are to be arranged in a manner to allow free circulation of air, door of oven should be opened after It cools down, that is, after 2 hours. Advantages and disadvantages. They do not require water, and there is not much pressure build up within the oven, unlike an autoclave, making them safer to work with. Suitable to be used in laboratory environment. They are much smaller than autoclaves but can still be as effective. As they use dry heat instead of moist heat, some organisms like prions may not be killed by them every time. Next we have moist heat. Moist heat can be categorized into three groups: temperature below 100 degrees Celsius, temperature at 100 degrees Celsius, and temperature above 100 degrees Celsius. First we have pasteurization of milk. First we have holding period, that is 63 degrees Celsius at 30 minutes, or 72 degrees Celsius for 15 to 20 minutes, followed by cooling quickly to 13 degrees Celsius or lower. This method is called flash method. This targets all the non-sporing pathogens, for example Mycobacterium, Brucella, Salmonella, Coxiella, Boutonii. A relatively high resistance may survive the holding method. Talking about the temperature at 100 degrees Celsius, boiling. Not recommended for sterilizing but used in disinfection. Sterilizing may be promoted by addition of 2% of sodium bicarbonate to the water. Holding period will be around 10 to 30 minutes. Steam at atmospheric pressure, that is around 100 degrees Celsius, used to sterilize cultural media. This is an inexpensive method. Holding period 100 degrees Celsius for 20 minutes in 20 minutes on 3 successive days. Talking about the principle, uh, kills first exposure kills vegetative bacteria, and then next exposure will kill vegetative bacteria that matures from the spores. Next we have steam under pressure, autoclave or steam sterilizer. Autoclave is a device that uses steam to sterilize equipment and other objects. This means that all bacteria, viruses, fungus, and spores are inactivated. However, prions may not be destroyed by autoclaving at typically 134 degrees Celsius for 3 minutes or 121 degrees Celsius for 15 minutes. Talking about the principle, water boils when its vapor pressure equals the surrounding atmosphere. Thus, when the pressure inside closed vessel is increased, the temperature at which water boils increases too. Holding period varies. Temperature between 108 degrees Celsius to 147 degrees Celsius. Items, dressing, instruments, laboratory wares, media, and pharmaceutical products. Talking about the several types of steam sterilizer: laboratory autoclaving, hospital dressing sterilizer, ball and instrument sterilizer, rapid cooling sterilizer. Even a domestic pressure cooker can be used as a sterilizer. Talking about the recommended temperature and duration, 121 degrees Celsius for 15 minutes, 126 degrees Celsius for 10 minutes, 134 degrees Celsius for 3 minutes. Disadvantages of autoclave. Some plastic ware melts in the high heat and the sharp instrument often become dull. Moreover, many chemicals break down during the sterilization process and oily substances can't be treated because they do not mix with water. Next we have filtration. Helps to remove bacteria from heat labile liquids, items, sera, and solution of sugars and antibiotics. Antibiotics. Talking about the principle, as viruses pass through the ordinary filters, filtration can be used to obtain bacteria-free filtrates of clinical samples. Type of filters: candle filters, asbestos filters, membrane filters, stringent glass filters. First we have candle filters used for purification of water for industrial and drinking purpose. They are manufactured under different grades of porosity. There are two types of candle filters: ungauged ceramic filters or diatomaceous filters. Next we have asbestos filters. These are disposable single-use discs trained to alkalize filter liquid. Use is discouraged because of its carcinogenic property. Next we have sintered glass filters, has low absorptive property and are expensive in use. Next we have membrane filters. Mostly these membrane filters are used and they are uh, more effective. Made up of cellulose esters or other polymers, usually used for water purification and analysis, sterilization and sterility testing, and uh, preparation of solutions for prepare. Next we have radiation. Two type of radiation: ionizing radiation and non-ionizing radiation. Talking about the non-ionizing radiation, infrared used for rapid mass sterilization of pre-packed items such as syringes, catheters. Next we have UV used for disinfecting enclosed areas such as entryways, operation theaters, and labs. Next we have ionizing radiation, firstly gamma radiation and X-rays, used for the sterilizing plastic syringes, swabs, athletes, animal feeds, cardboards, oil, grease, fabric, and metal foils. Talking about the testing efficiency of radiation, radiation control indicator disc. Special paper stick with stickers which is yellow in color is stick on the article. After proper sterilization, this color changes to red. Thank you. That's all we have in the first part.